Question number 1. Who was the first person to view cells? Hooke. Okay? Hooke was the first person to view cells. 1 mark for Hooke. Well, he's not Captain Hooke. I mean, it's Hooke. Well, that's easy for you to say. You're not Hooke. I'll let it go today, but you better spell it right in the future. It's not a hard word. It's not like anti-disestablishmentarianism, okay? Like, you should be able to spell that. Okay? Now, number 2. Who proposed the cell theory? These ones I might understand you spelling wrong. Okay? That would be Schwann with 2 N's and Schleiden, which actually is EI, not IE. Yeah, that one I could understand. That's an H. Sorry, it didn't measure. It didn't press hard enough. Okay? All right. That's just out of 1. They either got them both or they didn't get it. Okay? They don't get it. There were 2 people who proposed the cell theory. You get them both or it's wrong. Number 3. 3 points of the cell theory. 1 mark for all organisms are made of cells, or if they had all living things. Okay? 1 mark for that. Okay? Second one, cells carry out basic functions. And third thing, okay, is that cells come from the division of existing cells. Oops. One mark each for those. Okay. Number 4, when you're using a microscope, you always start with scanning power lens. Number 5 requires no background knowledge in microscopy to answer correctly. It only requires you to be able to find 25% of 400, which is 100 micrometers. They don't have units, it's wrong. 100 micrometers. You have to have UM, yeah. Number 6, I'll look it over. Number 6, what is the main reason we believe plants and animals have a common ancestor? They need to have mentioned essentially 2 things. Common structures, and if they listed a few, that's awesome. Okay? Common processes. Okay, and hopefully they mentioned a few of those too. They didn't necessarily have to, as long as they have something there about common structures and common processes. If they listed processes, they know what, then they were understanding that there were processes in common. Okay, okay, and that is number 6 is going to be out of 2. 1 for something about common structures and 1 for something about common processes. Functions is processes. Yep. The whole test, so this one was out of 2 and all the others were out of 1, so it's out of 7. No, sorry. No, it's out of 9. Yeah. Because there were 3 for number 3 and 2 for number 6. So, yeah, 9. Give them a mark out of 9, let them see it, and then put it up here on this desk. Okay, so yesterday we did all the pre-lab stuff for the microscopy lab, so that today we have basically over an hour to look at 4 different specimens. Okay, we want to have that kind of time because the first time you're using the microscope, things go a little bit slowly. Okay, and that's all right. We have lots and lots of time to work on it. So remember, the 4 things we're going to be looking at are the letter E, and in this order, the letter E, the amoeba, the paramecium, and then the euglena. Okay, those are the ones we're going to look at. Okay, we want to make a wet mound of the letter E. So there's some newspaper and some scissors up at the front table. Cut out 1 lowercase letter E from an article, not from a headline. Okay, put it on the slide, add water, put the cover slip on like I showed you yesterday. Okay, look at that underneath the microscope. Take a picture of it with your phone. Okay, make sure when you put the letter E into the microscope that it is right side up when it goes on the slide. That way you know what the microscope does to it in case it looks different through the lens. Okay, everybody follow me there? So it should be right side up, like you should be able to read it on the stage, then look through the microscope. Then after that, you clean off that glass rectangular slide, throw the cover slip and the letter E in the garbage, dry it off, and bring me back the glass slide. Then, grab an amoeba, take it back to your table, get it on scanning power, get it on low power, get it on high power as best you can. The letter E is probably the only one we'll only look at on scanning power. It does no good to blow up the letter E on high power because you won't see anything. Okay? You'll just see the fibers of the paper. The letter E will be blown up so big that you'll only see a little bit of it. Okay? So look at it just on scanning power. It'll take up pretty much the whole field of view on scanning power. Take a picture of it there. All the others you should look at, Kennedy, on high power. Okay, so make sure, guys, that everybody gets a chance, okay, to use the microscope. There's lots of microscopes, so this should be in like partners. So each partner gets to do 2 slides, okay, and get some experience using the microscope. If you're really struggling to get things focused, call me over and I can give you a hand. All right? So when we come back to the room, what we should have are pictures of the 4 specimens. Right? When we get back here, hopefully there will be time for me to show you how you can insert those into, or how you can make lab drawings from those. Basically, it's the smartest thing to do is print them off and then label them, okay? And then take a picture of them and insert that into your Google Doc. That is probably the quickest way to do it. Okay? Everybody follow. All right. Later. Okay, so to make the drawing, first thing you do is you go into Google Sheets, or sorry, not Google Sheets, Google Docs. Okay. You'll obviously do this in the template you have for your lab report, right? And what you do is you tell it you want to insert a drawing. And when you do that, it'll open up Google Drawing, which isn't a great like image editing program by any means, but it can draw straight lines and text boxes onto a picture, which is really all we need it to do. Okay, so one of the first things I do is go over here to Image, and I click on that, and I pick a picture file, okay, that I can use to, if it was my my lab, it would be one of the ones that was sent to me here. I'm just going to use the Nighthead here. All right, so it'll upload it and put it on there. Okay, I usually kind of make it take up the whole space. That way I got lots of room for my labels. Now I'm going to put in my text box that's going to be for my name and the date. Okay, so I'll put that up there, and I just go Mr. Coder. Whoop. All right, and then the date. So March, St. Patty's Day, March 17th, 2016. Okay, and then I can stretch that so it all fits on one line. There we go. And then I'm going to start doing my labels. Now, I don't expect that the labels on your drawings are going to be anything, you know, like that you would actually know what most of the parts are, because we haven't gone over that yet. But just put a couple of labels on, even if it's like inside of E, bottom of E, right? Okay, or little purple thing, all right? 'Cause, I mean, right now we don't know what all the structures are. As long as you got the format right, that's what I'm looking for right now. So for me, I would go, okay, I'm going to draw a horizontal line from this structure here, and I'm going to label this structure here, and I'll label this structure here. And then I'll just put, again, a text box, that's this thing here, and I'll draw it right here on the end of that line, and I'll just write that that's the plume, because that's what it is, okay? And then I'll insert another text box down here, okay? This will be the eye. And then another one down below, okay? This is the, like, shoulder pads or something, okay? Whatever, you put in the labels, okay? Then down below that, okay, so I click out of that and go back to the arrow, okay? I should be able to go a little bit lower down here and put in my other information like the title, size, and magnification that we showed before. So you're going to use text boxes and lines, and then when you're all done, you just click Save and Close, and it should insert it directly into your Google Doc, wherever you had your cursor at that particular time. Okay, so that's how you can do your lab drawings here. For this one, like let's say 3 or 4, okay, whatever you think you can confidently label or make up, I'm okay with. Okay, the next lab we do, you're going to have to accurately label cells, but at least that gives you an idea of how to do the lab drawings for now. Okay, and that's where you put the name of the thing you're looking at, so like amoeba or letter E, the size of it, which we went over yesterday, and the magnification. So for everything except the letter E, the magnification will be what? 400.